Good morning. My name is Marie Rienzo, and I want to welcome you to the NIH Office of Disease Prevention's Mind the Gap webinar series. This series explores research design, measurement, intervention, data analysis, and other methods of interest to prevention science. Our goal is to engage the prevention research community in thought-provoking discussions, to promote the use of the best available methods, and to support the development of better methods. Before we begin, I have some housekeeping items. To submit questions during the webinar, there are two options. First, you may submit questions via WebEx by clicking on the question mark in the WebEx toolbar. Please direct your questions to all panelists. Second, you may participate by Twitter and submit questions using the hashtag NIHMTG. At the conclusion of today's talk, we will open the floor to questions that have been submitted via WebEx and Twitter. Lastly, we would appreciate your feedback about today's webinar. Upon closing the WebEx meeting, you will be directed to a website to complete an evaluation. We would appreciate your feedback as it will help us improve this webinar series. At this time, I'd like to introduce Dr. David M. Murray, Associate Director of Prevention and Director of the Office of Disease Prevention.
Thank you, Marie. Today's speaker is Dr. John Quackenbush, who is a professor of biostatistics and computational biology at the Dana-Farber Cancer Institute and a professor of computational biology and bioinformatics at the Harvard School of Public Health. Dr. Quackenbush's research uses massive data from DNA sequencing and other assays to model functional networks in human cells. By comparing networks between groups of individuals, he's found new drug targets, explored chemotherapy resistance, and investigated differences between the sexes. He has made discoveries about how the genetic variants work together to determine our traits. Dr. Quackenbush has published more than 250 papers and has received numerous awards for his work, including recognition in 2013 as a White House Open Science Champion of Change. It's my pleasure to welcome Dr. Quackenbush.
So good morning. You can't see me this morning because I'm not able to share my screen, share my screen and my camera at the same time. But I wanted to welcome you, and I wanted to thank the organizers of this series for inviting me to share some of my work with you. So I've got 45 minutes, and I've got a pretty good story to tell. So I'm going to try to take you through that story as quickly as I can. And I'm going to tell a story about building models. And in that story, I'm going to challenge you to think differently than the way a lot of people think about models, and in particular about networks and cells. So I like to use quotes in my talk, and I'm going to open with a quote from George Box, which a lot of you have seen. Essentially, all models are wrong, but some models are useful. And I think this is fundamentally important when we think about biological systems. I'm someone who works hard to build models, and I recognize that those models are approximations of the truth. But the approximations we make are built by our understanding of how biological systems function. And I think that's really important. A lot of us think about tissue-specific genes or disease-specific genes. And I'm going to urge you to extend that model or that thinking a little bit to think about tissue-specific gene networks or disease-specific gene networks. And that's really the fundamental starting point for me today, is to explain how we approach the problem of building biological network models. So I'm going to imagine I have two different phenotypes, phenotype 1 and phenotype 2. And I'm going to measure some type of genomic data. And the type of data we find most useful, in large part because it's the most abundant, is data that we have on patterns of gene expression. Now, we have a long history in working in biology of taking that gene expression data, making contrast between phenotypes to identify differentially expressed genes, and to build models by taking those differentially expressed genes and, say, projecting them on a protein-protein interaction network. But here's how I'm going to challenge you to think differently. What we end up doing, and one of the things we find very informative, is to think about the genes as being fixed, but the connections between those genes, the edges in these models, being distinctive for each phenotype. So I'm going to infer phenotype-specific networks that are really defined by the patterns of connectivity that we see. And then we're going to take those models and we're going to analyze their topology and structure to see what that tells us about the underlying biological systems we're studying. We're going to look at differences in those network topologies and really focus on the edges that are different between different biological phenotypes. And then we're going to look at differential expression together with the changes in the structure to really understand what makes different phenotypes different by understanding the differences in their network structure. So what properties, what structures in the network can be linked back to differential activation of processes, and how can those processes help us understand the individual phenotypes? So I'll start with some assumptions that we use in model building. The first is that there is no single right network, that there are lots of networks representing really the array of phenotypes we want to try to understand. The second is that the structure of the network matters, and that network structure can change between states, maybe subtly, maybe profoundly, but we want to understand what those changes mean. So when we build networks, we recognize their approximations, and we are always challenged by people who ask us whether or not the networks we build are right. And I think that's the wrong question. I can tell you that they're not right, but the real question we want to ask is the network useful? Does it tell us something about the system that we're studying that we couldn't find another way? And the way we like to frame this question is does the network model inform our understanding of the underlying biology? Does it tell us something about the biological system that gives us new insight into how it functions? So my colleagues and I at Dana-Farber and Harvard have been working on building models for a number of years, and we've built a wide range of different models depending on the type of data that we have available. In my group and in our collective group here, we tend to name them after animals, so we refer to this as the Methodological Zoo. And we've really thought carefully about the different data input types that we have, the type of information that's available, and we adapt our modeling strategy to take into account different types of data. The reason why we don't have a unified framework yet is largely because we haven't had a single data set that encompasses all the different data types we'd like to try to build. But I'm going to talk about at least two of these modeling approaches today. The first is Condor, and Condor is a method that we use to try to model gene regulatory interactions that occur by looking, or that we can observe by looking at expression quantitative trait loci. The second is a set of models we build around gene expression data that we use to try to model gene regulatory networks. And if I have time, I'm going to talk about LIONESS, an approach we use to extrapolate those models to individual sample networks. So I don't have time to take you through all of these today, but all of the methods are available. They've all been published either in journal papers or on the archive or bioRxiv. All of them have software code, and all of them are available for use. But I'm going to start by telling you this story about modeling. And I'm going to start by looking at a really fundamental question in understanding biological systems. Can we solve the puzzle of GWAS? So what do I mean by this? Well, most of us today are familiar with genome-wide association studies. We take DNA from individuals, we sequence that DNA, we look for genetic variants at a single nucleotide polymorphism or a SNP, and then we compare populations, say a healthy population and one with a disease, and we look for a higher incidence of a particular SNP within that disease population, with the understanding that if we see a profound prevalence of a particular variant in that disease population, that variant is likely to be associated with the phenotype we're observing, in this case, the disease. And so if we have a large enough population, we can actually find significant association. So this is a really interesting approach to looking at disease, and it's very widely used. But disease is just an example of a complex phenotype. So there have been some interesting studies that have tried to look for genetic basis of complex phenotype. And part of the reason they've done this is that many studies of disease haven't found a large number of genetic smoking guns. They find genetic variants that have small effect sizes. And this is an example of a study that was published about 2.5 years ago, looking at a common variant, which we know has a genetic component or a common trait, human height. And in this case, this group looked at about 250,000 individuals. And what they discovered is they could explain 20% of human height using about 700 genetic variants. To get to 21%, they needed almost 2,000 variants. To get to 24%, almost 4,000. To get to 29%, almost 10,000, right? 10,000 genetic variants to explain something that we know clearly has a genetic component. So, you know, what's the challenge? What's the problem? Well, a lot of these variants have very, very, very small effect sizes. The same group then looked at body mass index, and in this case, they looked at almost 340,000 individuals. And when they did this, what they discovered was that 97 SNPs would explain about 3% of BMI, and that in fact, to get to explaining the vast majority of body mass index, one needed almost all common SNPs. And that in fact, all those SNPs would explain only about 20% of body mass index. So the question we have to ask ourselves is, given the failure of GWAS to find these strong genetic variants, what do? Well, one of the ideas that's been advanced is that we can simply find map everything, that we can look for rare genetic variants that might carry disease phenotypes. And there's a very nice paper published about 1.5 years ago looking at diabetes. This group looked at 111,000 individuals. And their conclusion was that these lower frequency variants don't play a major role in predisposition for type 2 diabetes. And there's a nice little graph that appears in what I believe is figure 1, showing the empirical data on the left and fitting different models. And the bottom line from this analysis was that these rare variants simply don't explain disease risk. So I and my colleagues started to ask ourselves, how can we actually look at genetic variants and place them in a biological context so that we can start to understand maybe how these genetic variants interact together to influence phenotypes, given the fact that common variants don't seem in general to have large effect sizes. What that suggested to us is they had to work in concert. So to start this process, we went back to an old idea, looking at what's called eQTLs, or expression quantitative trait loci. Now, eQTL analysis is really a variation on an old idea, quantitative trait locus analysis, developed by Steve Tanksley in the 1970s. And Steve was looking at an agricultural application. He was trying to do quantitative breeding of tomatoes using genetic data. And so what Steve realized you could do is look at genetic variants and look at quantitative traits like the redness or the roundness or the density of tomatoes, and look for associations between the genotype in a particular locus and the strength of that quantitative trait. So the idea behind eQTL analysis, which was developed about 16 or 17 years ago, was to take that idea of quantitative traits and extend it to all 25,000 genes in the genome and their expression levels. So eQTL analysis uses genome-wide data on genetic variants with expression data. It treats gene expression as a quantitative trait. And we simply ask which SNPs are correlated with the degree of gene expression. Now, one of the things you see if you look at papers on eQTL analysis is that most of those analyses concentrate on cis-acting SNPs. Those are the SNPs that are immediately adjacent to the gene. But one of the things we started to ask ourselves in looking at this analysis was, how do we account for what are called trans-acting SNPs, SNPs that are at a great distance on a genome scale from the individual gene, not those SNPs which are possibly modifying a promoter, but those that are far away. So the idea between looking at cis-acting and trans-acting SNPs is really pretty simple. If we look at cis-acting SNPs and do an eQTL analysis, we can look at a particular locus adjacent to a gene. We can see if we have the common variant. In this case, you can imagine having 2 Cs. The rare variant, 2 Gs, or a heterozygous genotype, a C and a G. And we simply look for genes whose expression levels are correlated with the genotype in a linear way. Right? We can also look at trans-acting eQTLs, and in this case, we're looking at a gene, but not SNPs that are immediately adjacent to it, SNPs that are somewhere else in the genome, where we can look at the genotype and simply ask, is there a correlation of a distant genotype with the expression level of a particular gene? So we have cis and trans-acting SNPs, and what we wanted to do was to look at a large data set to try to identify these types of associations. So we took a data set that was available from the Lung Genomics Research Consortium, LGRC. It was a group of individuals who had lung tissue profiled for gene expression. There was also genotyping data. And we did a standard eQTL analysis using a widely available tool called Matrix eQTL. We ran that eQTL analysis, and we took that data that had genes and their associated SNPs that were where the genotype was correlated with the pattern of expression. And the trick we used was we represented that data as what's called a bipartite graph. We represented it as a network. So what's a bipartite graph? The idea is very simple. A bipartite graph has 2 types of nodes. You think of them as squares and circles, or SNPs and genes, and we represent an edge, a connection between them, if there's a significant association between the SNP genotype at a particular locus and the expression of a particular gene. So you can imagine from looking at 6 million SNPs and 25,000 genes, we have lots of many-to-many connections. So we can have one SNP that seems to influence many genes, or one gene that seems to be influenced by many SNPs. And the unique thing about this kind of graph is that we don't have associations between SNPs and SNPs, or genes and genes, only between the 2 types of objects. So we're going to draw this graph, and we're going to ask, can this set of associations actually inform our understanding of how these SNPs are influencing expression and ultimately altering function in this model of chronic obstructive pulmonary disease? So every time you see a network talk, somebody has to produce a graph like this, a hairball showing all the connections. In fact, we don't find these graphs to be very useful. And this isn't a graph that I put together. This is actually just some random hairball I grabbed off the internet, because these graphs, as a big representation of the data, don't provide us with a lot of insight. But you can take these graphs and you can actually look at some of the properties. And one of the most important properties we find in looking at networks is what's called the degree distribution. The degree is simply the number of connections that a node, in this case a SNP or a gene, in the network has. So we can have highly connected variants or less frequently connected variants, and the frequency is simply the number of times that we see a particular level of connectivity. So I'm going to focus on the degree distribution for the SNPs, and this just shows the same graph I had on the left-hand side on the previous figure. And what you can see is the kind of distribution that we characteristically see in naturally evolving networks. So this is very much like the internet. If you look at the lower right-hand corner, what you'll see are what we call the hubs, those things that are highly connected. In the upper left-hand corner, we see the leaves. If you make a log-log plot like this, what you see for these naturally evolving networks is a straight line. People call this a scale-free distribution. In fact, formally, it's a fat-tailed distribution. But this is like the internet. Up here is your laptop, and there are lots of laptops out there. Down here are the hubs, like Google and Amazon and eBay. And the difference between these is that if you think about the hubs, right, if Google or Amazon shut down, everybody would freak out. But if your laptop turns off, probably nobody notices except maybe your friends and family. So down here are the hubs, which are really important. Up here are the leaves. And one of the questions we asked ourselves was when we made this graph in chronic obstructive pulmonary disease for lung tissue, if we looked at disease-associated genetic variants, either in COPD or in all diseases, where do they map? And the interesting thing we saw was that while we expected to see some things hitting the hubs, what we really saw, and hopefully you can see little red lines, little red dots appearing in the upper left-hand corner, but if you look at this, what you see is that the hubs are actually a GWAS desert, and all the genetic variants appear in this upper left-hand corner. The other thing which you might notice is that the leaves are actually a little bit underrepresented, and the kind of middle of this graph is overrepresented. So we were really curious about that because we thought it might lead us to some kind of internal structure in the network. So we wanted to ask ourselves whether or not we could look at the properties of this network to understand what these SNPs do. And the idea we developed was to take this network and look for communities based on the structure of the network. Now, it turns out that most networks have communities, and there are lots of ways of identifying these communities. A lot of them are based on the idea of maximizing modularity. If you want to think about communities in a very natural way, think about the fact that you have a mobile phone, and you can call anybody in the world, but you don't, right? If you're married, you call your wife or husband, your spouse calls your son calls your daughter calls her grandmother, your mother calls you, right? You have a family network that's represented by an increased likelihood that on any given day, any two of you are going to call each other relative to calling a random person on the outside, right? So while you may one day call me because you think this presentation is brilliant and want to talk about it more, right, your mother is never going to call me. So you have a family network that is a family community is defined by that increased likelihood that any two of you are going to call each other. In the same way, you exist in other communities. You have a work community, you have a social community, and in fact, if I could map the community structure of telecommunications, I could probably predict whether it's a work day and you're calling your friends, or a weekend, or sorry, work day and you're calling your colleagues, weekend you're calling your friends, a holiday and you're calling your family. Right? So we can apply that same sort of idea to looking at these biological communities, biological networks. And we can identify communities by using well-established methods that look for community structure. And the interesting thing we saw in our COPD data is that when we looked at that data, we saw, in fact, very, very strong, highly modular communities. So this is a representation of the network using what we call a degree distribution. And sorry, not a degree distribution, an adjacency matrix. And in this matrix, I'm simply representing ones and zeros, or dots and blanks. A dot appears if there's a connection. And what we have is that each row represents one of the genes in our model. Each column represents a SNP. And what you can see is that this community actually organizes into very, very rich, very strong communities. Now, if you look, you see vertical stripes, and those are really the global hubs. But John Platek, who worked with me, came up with a different representation of the network. And I like to think about this as sort of Botticelli's Venus. You've got these little seashells with beauty coming out, but these are the individual communities. And what you can see is the communities themselves have structure. The interesting thing is when we looked at these highly modular communities in the eQTL network, if we focus on the giant connected component, all those things that are connected together, not the little satellites shown in the lower left, but if we look at this giant connected component, what we see is that many of these communities are strongly enriched for particular biological functions based on co-term analysis. And this is really sort of interesting. We started our analysis with the old GWAS approach. We think about single genes being associated with single traits, or eQTL, single variants being associated with the expression levels of individual genes. And what this model tells us is that families of variants work together to influence the expression of individual functions. Now, the interesting thing is when we started to map disease-associated SNPs in COPD with into these different modules, what we discovered is they map to a small number of modules, 3, and that those modules had functions which were associated with what we know about the development and progression of COPD. So we're starting to gain a really interesting understanding from this very simple model that genetic variants which are associated with a particular disease map to communities where their presence may perturb the expression and therefore the function of those communities, which is exactly what we should expect for these common genetic traits. One of the other ideas that John Plattig, postdoc working with me, had was that when we look at these communities, if you look at those seashell plots, you can kind of see that there are local hubs in those communities. So in working with John, we identified what we call the core score, and that's basically the fraction of the modularity which is carried by an individual gene. And so we wanted to ask ourselves, what can we learn from looking at these individual core scores? And the interesting thing is that when we took 34 COPD-associated SNPs from GWAS studies, and we looked at their core scores, what we found was that the core scores of those genes were more than 20 times higher than the average core score for the other SNPs in the network. And this is really interesting because what it says is that the disease-associated genetic variants aren't randomly distributed across the network. They're concentrated in communities, and in fact, they're central to their communities. They hold those communities together. So the way we think about this is that a disease-associated variant is not just any random variant. It's a variant that's most likely to perturb a function which is related to the development and progression of that disease. And that's kind of a fascinating discovery about what really drives genetic-associated diseases and how all these small effect variants can work together to influence function. So we published an analysis of this about 2 years ago in PLOS Computational Biology. But then we started to ask ourselves just how general this overall finding is. So we had a follow-up paper that appeared at the end of last year in PNAS, and in that we looked at data from one of the most interesting publicly available studies, the GTEx study, the Genotype-Tissue Expression project. In this project, they've collected data from autopsy samples. We worked with Release 6 that had about 600 autopsy samples. For those individuals, they genotyped them all. They also biopsied as many as 50 different sites around the body and generated RNA-seq gene expression data. So we took those data, we took the SNP genotypes, we had 13 tissues in which we had enough data to actually look at eQTL analysis, and what we discovered was in each of those communities, we saw exactly the same thing we had seen previously in the lung tissue eQTL data, that those eQTL networks are organized into highly modular communities. So here are the different tissues showing the modularity. And if we look, for example, at heart left ventricle, what we see is exactly that same kind of structure. More interesting, I think, is that when we look at things like the degree distribution shown in the upper left, what we see, and it's a little bit difficult maybe to see, but you see blue dots here, which are GWAS hits. Those blue dots appear in the degree distribution overrepresented in essentially the same way we saw for COPD. And Maud Fagny, who did this analysis, actually has a better way of showing it. This is the observed number of GWAS hits versus expected. And what you see is there's kind of a cliff around degree 10, which is interesting. If you have more than about 10 connections, you don't appear in any GWAS studies. And we think that that may be because SNPs with too many connections, if they have a deleterious variant, are simply swept out of the population. When we look at the core scores on the right for GWAS versus non-GWAS hits, what we see, looking at all GWAS hits in the GWAS catalog, is that those SNPs, which have been identified with physical traits like diseases, have a significantly higher core score. And we see exactly the same thing looking at all 13 tissues just picking out a simple group at random, showing you the expected versus observed plot for where we see these GWAS hits. So we're starting to see something really interesting emerge from this. What's even more interesting, though, is we start to delve into the function of these SNPs. What we discover is that core SNPs are not only more likely to be associated with disease, they're more likely to be functionally annotated. And when we cross-reference this data with data from the Roadmap Epigenomics Project, what we see is that core SNPs are different. And in fact, when we look at these core SNPs, what we start to see is that we have tissue-specific functions that are organized around SNPs that fall into tissue-specific open chromatin, that they're unique communities in each tissue, but they nucleate around SNPs, core SNPs, that in fact map to open chromatin, which are only open and accessible in that individual tissue. So this actually paints a really interesting picture, and I just recognized my animation is out of order. But if we look at this, what we see is that in general, the global SNPs are not GWAS hits. In fact, GWAS hits map to cores of communities, and that those individual communities have specific functions that are either associated with individual tissues or with biological processes, which are relevant for understanding the phenotype. So from a very simple model, while I can't tell you that any single edge or any single association is true, the overall patterns that we see in this model have a great deal of explanatory power. So that's story number one that I wanted to tell. The second story I wanted to tell is one about modeling gene regulatory processes. So this builds around the second method I mentioned earlier, PANDA. PANDA is a way of modeling gene regulation based on starting with what we know about transcription. So if I look at RNA-seq data or microarray data, what I'm going to do is measure the presence or absence and, in fact, the degree of expression of individual RNAs. To make these RNAs, what I need to do is assemble an RNA polymerase construct, and what we know is that's typically facilitated by the presence of individual transcription factors that bind to the DNA, that open the chromatin, that help with the assembly of this transcription factor complex, this transcriptional complex. So what I'm going to do is I'm going to think about this. I'm going to try to model this using a very simple idea from communications theory called message passing or affinity propagation. So I'm going to imagine I have a transcription factor, and it's going to send a message to its downstream target. It's going to tell a gene to turn on or turn off. And when we model this communications process, we're going to recognize that for any edge in this network, there actually have to be two active participants for information to flow. The transmitter has got to transmit, but the receiver has got to receive. And the example I always give is when I come home at the end of the day, I walk in the door, and my wife is excited to see me, so she starts telling me about her day. But in the first few minutes before I set down my bag or take off my coat or take off my shoes, I don't hear anything she says, right? So even though she's transmitting, I'm not receiving. Right? If my son were in the room and he heard our conversation, he could actually tell you whether she told me something or and I didn't hear it, or whether she failed to tell me. But with just two of us, there's no way to really know without hearing our conversation where that communication broke down. On the other hand, if my son is there or we have other individuals, what we can start to do is we can start to model this process and iteratively estimate where communication breaks down. So on each edge, we define two functions. One we call the responsibility, and the other that we call the availability. And what we do is we estimate these based on our understanding of the system we're modeling. So we're going to start with a guess as to what this network looks like. We're going to take transcription factors and we're going to map their binding sites to the genome and build a preliminary network. Then we're going to take other data types, gene expression data, and also, if it's available, protein-protein interaction data, because we know transcription factors form complexes. And we're going to take that information, we're going to update the model by estimating these functions by looking at correlations in the overall network structure. We're going to update that network, and we're going to iterate this process until the network converges. And the idea is actually really simple. We're trying to maximize the information flow through the network. So we've applied this in a lot of different settings, and I'm just going to give you one example that we published a short time ago, looking at two subtypes of ovarian cancer. And these subtypes are distinguished based on the expression of genes associated with angiogenesis or the lack of the expression of those genes. So we characterize the ovarian cancer subtypes as being either angiogenic or non-angiogenic. And what we did is we took this basic classification system and we extended it to the TCGA ovarian cancer data. So we had two groups of patients. We had those with the angiogenic subtype based on this classification and those with the non-angiogenic subtype. We took the motif data to estimate an initial network. We took protein-protein interaction data with the gene expression data, and we built individual network models for each one of the subtypes. And then we compared those networks and their structures, and we asked, what are the differences? And this is one of the very first applications of this method to a large-scale biological system. And when we looked at this, one of the key things we recognized, which is what I alluded to when I talked about networks earlier, was that it's not the nodes, but the edges that are the atom of the network. So I'm going to show you a representation of the network for the angiogenic and non-angiogenic subtypes overlaid on top of each other. The angiogenic is in red, the non-angiogenic is in blue. There are 10 transcription factors which have the biggest differences between these two networks in terms of their connectivity. So they're the gray circles in the middle. And if you compare these, here's the angiogenic, here's the non-angiogenic. If I can compare them, you see that there are differences. And it's usually easiest to look around the periphery of the network, but there are differences between these networks that you can start to pick out. There are different patterns of communication. So Kimberly Glass, who worked with me on this, actually came up with a different way of representing this. I apologize you can't read the names of the transcription factors, but they're arrayed around the center. And around the outside of this spirograph representation are all the genes which are differentially regulated in our model. They have edges that differ between the two subtypes. They're all the differentially regulated edges. And what's interesting is as we started to look at these differential patterns of regulation, what we saw, like on the left-hand side, was that there were some very simple sets of connections, but sort of like down here in the lower right-hand corner, there's some places where there are multiple transcription factors which seem to work together. And when we analyzed these data, what we discovered was that those transcription factors which seem to co-regulate particular genes occurred far more frequently than we'd expect by chance. In fact, if you look at the p-values here in the center at the top, those p-values are just way off the charts in terms of their significance for the overrepresentation of these individual transcription factors. So as we started to tease this apart, what we recognized was that those transcription factors are actually known to form complexes, and that those complexes are associated with processes involved in angiogenesis. So this network model of differential regulation identified through building these PANDA gene regulatory networks, one for angiogenic subtype, one for the non-angiogenic subtype, and comparing those two individual networks leads us to identify sets of transcription factors which work cooperatively to influence angiogenesis. And in fact, there's evidence published that shows that if you disrupt the formation of these complexes, you can actually interrupt or interfere with the process of angiogenesis. So the network models actually tell us a great deal about what drives these systems. So we published this in BMC Bioinformatics, and we've since published a number of other papers looking at various applications of PANDA to identify differences in the regulatory processes and in the network structure between different individuals with different disease subtypes. We published at the end of last year what I think is a really nice paper in Cell Reports that takes this model and extends it now to looking at 38 different tissues. And one of the fascinating things we see is when we look at these models across different tissues, there are actually core sets of processes which are almost universal, but we see tissue-specific functions emerging through changes in the regulatory processes which exist in these underlying networks. So the networks and their structure tells us something fundamental about the process. In the final minutes I have, I want to take you, though, to address one other really fundamental question, and that is, can we move beyond the network? Now, I've already told you that different phenotypes have different networks, but if you think about all the people listening to this webinar, everyone here, in any tissue we look at, assuming we're all healthy, we all express genes differently. And I would argue, based on our understanding of these PANDA models, that each one of us should be represented by a different individual gene regulatory network. So we started thinking about this problem, right, that we take a group of samples, and from that group, whatever network reconstruction method we choose to use, we estimate one network. And that's really an aggregate network for this population. But we started to think, what if we take all of the individual samples and try to understand how each sample contributes edge by edge, connection by connection to the network. And what we developed is a simple approach based on linear interpolation, right? The same kind of interpolation you learned in high school math to try to estimate complex functions by using a linear approximation. We could take that same basic idea and apply it to learn individual sample networks. So this is really work done by Marike Kujer and Matt Tung. Matt was a post-bac student who worked with us. And what Matt did was he looked at a population and said, all right, I'm going to construct a network for that population. And the problem we assigned Matt was to look for ways to estimate variations in each one of the edges in these networks. So the natural idea was to do a bootstrap analysis where we leave out some samples, but Matt chose to leave out just one sample. And what happened was, when he did this, he recognized the network with that one sample and without that one sample, edge by edge, were subtly different in the estimates of edge strength. So as we talked this through, what we realized is we subtracted the network with that sample from the network without that sample, we could get an estimate of this sample's, that left out sample's, contribution edge by edge to the network. And if we took that and simply scaled it and added it back to the n-1 network, we could get an estimate of the network for each individual sample. And so this is really a revelation. The mathematics behind it is very, very simple. It's simple linear algebra on matrices, where the matrices represent the estimated networks that come from a method like PANDA. So we wanted to test this, and in the paper that we published, or that we have on the archive about this method, we actually took yeast cell cycle data. This is the Paul Spellman data, and we estimated for each one of the two replicates individual sample networks. We all know the gene expression data is correlated, but when we looked at the individual edge weights, what we saw was that between the two replicates, the edge weights were also correlated. If we took those two replicates and put them time point by time point next to each other, what we could start to see is what we'd hope to see, that the edge weights vary over time. So this is showing a heat map where each column is a different time point, and each row is an individual gene, an individual edge, not an individual gene. And we can see the edge weights, the 1,000 most variable edge weights, oscillating over time. When we looked at those edge weights, there are actually 4 transcription factors that were highly represented, all of which are known to be associated with the cell cycle. And when we looked across the edges of their targets, what we found was that their target genes also oscillated. So these were networks built with PANDA. And if you look at the right-hand column, what you can see In red is the oscillation of the edge weights, the oscillation of the gene expression levels for the target genes. And in black, you can see the oscillation of the edge weights. Now, when we first saw this, we started to notice the edge weights in black were oscillating with twice the frequency of the target genes. But then we realized this was what we should have expected, that this was a model from PANDA, and what PANDA's model was telling us was that a message is being sent to say, turn on, turn off, turn on, turn off, turn on, right? The model is telling us something fundamental about the regulatory process, in this case associated with the cell cycle. When we look at simple metrics like mapping these back to look at transcription factor occupancy around these target genes, what we find is that PANDA with LIONESS actually does a better job of estimating where those transcription factors bind at each individual time point than any other method we've been able to test. So this is fine for yeast, but you might ask, well, what does this tell us about human biology? And the last example I want to give you is just a really simple one. This is work done by Marieke Kujer. Marieke took LIONESS and she applied it to an analysis of samples for glioblastoma available through the TCGA. So she took the group of patients and she divided them into two groups, poor prognosis and good prognosis. And it was just defined very simply based on 3-year survival. She then used LIONESS and PANDA to estimate networks for each individual in this population. And she treated the edge weights as a derived measure on the population. And she asked, are there significant differences in these regulatory connections between the good prognosis and the poor prognosis group? To do that, she used a tool that many of you have probably used for analyzing gene expression data, LIMA, but instead of using gene expression as an input, she used edge scores. And what she found were important roles for edges that were initiated either at FOS-June or at NF-kappa-B, and that when you looked at the targets for those genes, what you start to see is a differential targeting in good and poor prognosis. Then when we look at the functions associated with that targeting, what we start to see are processes like mitosis associated with poor prognosis and immune response associated with a good prognosis in patients. So what we've been able to do in looking at gene expression is to understand that gene regulatory networks are a way of defining phenotype. That if we compare gene regulatory networks between phenotype, we see differences in connectivity that give us insight into the underlying biology. If we use an approach that allows us to tease out individual gene regulatory networks for each person in a population, we can actually look at differences in those networks, look at differences in the connectivity, and by analyzing those regulatory differences, which were derived from the model, actually get insight into the drivers of disease and response. So I hope in the time I've had, I've been able to show you that while networks may not solve everything and networks may not be perfect, these networks can actually provide a framework for us to interpret, in the first case, genetic variants and their associated with phenotype. In the second case, regulatory patterns and their associations with phenotype. And I can tell you that the things we've learned from both of these models are biologically sensible, give us insight into the biology, and they're things that we could not have found by simply looking at differential expression. So I've used my time. I like to use quotes, so I want to close with a quote. This is one of my favorite quotes. This is from Enrico Fermi, who's a physicist. Fermi said, "Before I came here, I was confused about this subject. After listening to your lecture, I am still confused, but at a higher level." So I hope if you're confused, it's highly confused, but I'm really pleased to have a few minutes to be able to answer questions. So I just want to close with an acknowledgement slide, and I want to thank you all for taking time and spending time with me here today. So I'm going to end sharing, and we'll see if there are any questions.
Thank you, John, very much. We do have questions. One of the things that you started with was that all models are wrong, but some are useful. Networks are like models, and all of them are wrong, but some of them are useful. How do you know when you've gotten a network that's useful?
So it really comes down to the interpretation. And I talked about, our models are these networks. The networks are our models. And the question we always ask is whether or not the network model itself has some explanatory power. So for the eQTLs, what was really fascinating for us is that when we looked at these SNPs that were found through GWAS, all of which had small effect size, none of which would explain the individual variation or the individual traits in a really clear way, we saw them clustered around functions. And it allowed us to do two things. Number one was to take SNPs, some of which had no really clear function in the disease, and map them to a functional context, which made sense. The other was to understand how these variants could work together with small effect sizes to amplify their size, right? I think about it as sort of the Lilliputians tying down Gulliver, right? Each individual rope they threw over him couldn't hold him down. But if you throw thousands of ropes over him, he can't move. In the same way, all these genetic variants operating individually don't do much, but in communities, they can actually shift functions, and that shift in function can be tied to disease. So there, the analysis was really the integrative effects that told us we were onto something. In the modeling of gene regulatory networks, what's been fascinating in case after case is that when we build these regulatory network models, we find that the regulators and the patterns of regulation take us a long way to understanding what's driving the biology. So Camila Lopez-Ramos, who works with me, now has a paper. It's on the bioarchive. It's been submitted to Cancer Research that looks at sexual dimorphism in colon cancer. And men and women have a different natural history of disease. In looking at differential expression between men and women, we don't see a lot that tells us why that is. And looking at differential regulation, probably not surprisingly, we see different patterns of regulation around genes that respond to estrogen and testosterone. They have hormone regulatory elements. And the modeling gets us to an understanding of disease that makes sense that we couldn't have found by simply looking at differential expression. So time and again, these models are taking us incrementally closer to what I think is biological insight. So I see a question on my screen. I don't know if anybody's still there. But one of the questions is whether the cooperative transcription factors are master regulators. Oops, I'm scrolling through a whole bunch. Are master regulators or not? And that's a great question. The interesting thing is that they're not always seen as master regulators. When we looked at 38 tissues from the GTEx data, what we really saw was that the transcription factors that were driving tissue specificity weren't things that were to the core of the network, that the core of the network itself was in many ways almost immutable. It was basic metabolism. It's what you might think of as housekeeping functions. But as we started to look for where real differences were in the network, it was sort of at the periphery. It wasn't things way on the outside. It was sort of at that intermediate level. And what we saw was when transcription factors became tissue-specific and started to regulate tissue-specific processes, they gained more connections. And they actually gained more paths between them. Their betweenness went up. And what that suggested to us is that the shifts in phenotype were associated with shifts in regulatory processes. So I'm not sure what the protocol is, if I should continue to read questions or
John, I was going to draw your attention to another one of the questions.
Okay, good.
Someone has suggested that you've been presenting data from retrospective analyses and wondering, if you ever do prospective studies to try to confirm the models that you're finding in the retrospective work.
So again, I'll sort of group this into two classes. For the eQTLs, we haven't yet done that. We've been working on trying to develop good ways of making predictions from the SNPs, how they're going to alter the phenotypes. And we have some ideas based around looking at collections of SNPs, possibly weighting them by their core scores, and coming up with an aggregate score. But the challenge that we face is that for the SNPs, each one of the SNPs has its own really small effect size. So it's hard to try to understand how we make the right kind of perturbation from lots of individual small effect size SNPs to affect the phenotype. I think one of the best things we can do with the eQTL networks is to try to understand how effective they are in predicting phenotypes by looking at populations. And that's something that we're just really starting to try to do. For the gene regulatory network modeling, in fact, we have done experiments. And so there's a nice collaborative paper we published with Keelan Tenestra looking at the effects of gene regulatory networks in asthma in response to corticosteroids. And Keelan has actually done a really nice set of laboratory experiments that follow up on the predictions of the model and actually validate a lot of the predictions that come out of the model. So, the models don't exist in isolation. If we can't make predictions that can be validated in the lab, we have trouble believing them. And, in asthma and chronic obstructive pulmonary disease and other applications, we're actually doing laboratory experiments that follow from the identification of what we've seen in these models and that provide validation for the models themselves.
Do we have any examples where this kind of modeling has led to a new therapy, a new treatment?
Not yet, but that's one of the things that we hope it does get us to. And we've been working very closely with some collaborators, Ed Silverman, Don DeMeo, Pete Castaldi, Craig Hirsch, working on chronic obstructive pulmonary disease. And, we've actually found some very interesting things that members of that group are following up in the lab to try to see whether or not they can move us in a therapeutic direction. Marika Kucher's also been working on applications in different cancers. And again, we have some early hints about interventions that may have an influence on disease and disease response. But to translate those to potential therapies takes a lot of additional work. So we're at the start, but it's a long journey.
Great. Thank you very much. We're clearly not going to have enough time to answer all the questions that are rolling in, many of them quite technical. And what I hope we can do is send you the questions that we've not been able to address, ask you to prepare brief responses, and then we can post them on the website along with the other materials associated with the presentation. I want to thank you very much. Go ahead.
I'd be happy to, and I'd welcome anybody who has questions to drop me an email. I'm pretty easy to find. So thank you very much for hosting this, and thanks everybody for joining and for sending questions.
Thank you, John.
Thank you, Dr. Murray, and thank you to everyone who participated in today's webinar. We will be posting a recording of today's webinar on our website, prevention.nih.gov/mindthegap, in the next few weeks. You will receive an email with a link to the recording when it is available. Thank you.